Hi, everybody. This is Dr. David Ross presenting the conference today. First, I want to thank the VNI and Randy and Karen for helping to organize this and helping to set up this webinar today. I'm really looking forward to this opportunity. The topic is measuring MRI brain volume in patients with chronic traumatic brain injury, mild to moderate traumatic brain injury. A little bit about myself. I'm a neuropsychiatrist and a brain injury physician. I have an outpatient clinic called Virginia Institute of Neuropsychiatry in Midlothian, Virginia. And we do not only a lot of clinical treatment, we do a lot of forensic work, and we do research and development as well. And today I wanted to talk to you about our latest research. So it's nice to be able to communicate with, I'm sure, a lot of my academic colleagues as well, which I don't get to do as often as I'd like to. I'm going to switch it over now to the slides. And you can see the title, which is basically the summary of the whole talk, which is that patients with mild or moderate traumatic brain injury have abnormal brain enlargement. By way of disclosure, let's see. I'm the CEO of NeuroGage LLC. That's the company that makes NeuroGage software I'll be talking about today. In 2017, I was a paid consultant for CortechsLabs, which makes NeuroQuant, also software I'll be talking about today. NeuroGage is not FDA cleared. That's our own software. It is based on NeuroQuant, which is FDA cleared for measuring MRI brain volume. And I'll tell you more about NeuroQuant during this talk. And although NeuroGage is not FDA cleared, we do have about a dozen articles or so now in the peer-reviewed literature supporting its reliability and validity. Okay, this talk has a couple twists and turns to it, so I want to go ahead and tell you right at the start what the main conclusions will be. I'm hoping that'll help you follow along. First of all, we measured MRI brain volume in our patients with chronic mild or moderate traumatic brain injury. Our patients here at our clinic typically are in car accidents or maybe slip and fall at work, or something fell off a shelf, hit them in the head. We don't have military population. We have very few sports patients, although it's an area we're working on getting into. These are mostly accident type patients. They're all chronic. They've had symptoms, neuropsychiatric symptoms of many types, cognitive symptoms, impaired sleep and wakefulness, chronic pain, mood problems, etc., for usually months and often years after their injury. Otherwise, they don't come to our clinic for evaluation and treatment. So that's the kind of patients and what we did. And we measured the volume, and we found that patients had abnormal brain enlargement of many regions. They had atrophy or shrinking of some regions, but not as many as abnormally large. And they also had a lot of abnormal brain asymmetry, one side of the brain bigger than you'd expect based on the side of the other. And toward the end, we'll talk about a hypothesis relating the abnormal enlargement and asymmetry that we found. So by way of background, everybody knows that traumatic brain injury causes brain atrophy. Here's some general reviews I listed down here, and we've been researching this for over 30 years, and there are many, many dozen studies showing that traumatic brain injury causes atrophy or shrinking of the brain. And so, you know, I'm going to have to do some convincing for you to believe that we actually saw a lot of enlargement in our patients. One thing to remember, most of these earlier studies were done in patients with moderate to severe traumatic brain injury, where you can just look at an MRI or a CT scan and see the brain is shrunken and the ventricles have gotten larger. But there aren't as many studies in chronic mild to moderate TBI, so things could be a little different there. So that's part of what we're dealing with. And my patients tend to deal with that issue. They often come here, if you just talk to them casually, they look and sound kind of normal at first glance. Some people aren't really sure they have such chronic, terrible symptoms that they claim to have. Some people don't believe them. And if you look at their MRI, it often looks normal, just one typical visual inspection. And so, you know, several years ago, we started measuring the brain volume in these patients, and we found a lot of volume abnormalities, which provides objective evidence of injury, which can really help vindicate these patients. So that was the context in which we did this study. And we mentioned that. And so NeuroQuant, which is what we've used to measure brain volume all these years, was a major breakthrough. And it was cleared for clinical use in 2006 to, as I think I mentioned earlier, identify brain regions on MRI and measure MRI brain volume. Because up until that time, the MRI brain volume measurement was confined to research academic settings. You know, if I wanted to order a brain volume study on my patient, I couldn't do it. But since NeuroQuant was approved and marketed, we can do that. There are a couple other software programs since then that have also been marketed and approved for use. And we mentioned that. And to summarize a lot of literature, there are about 60 peer-reviewed published articles on NeuroQuant now. There have been over 100,000 people who's had brain volume measured with NeuroQuant. And I'm just going to summarize all that and say that in my opinion, and probably the opinion of other people who work in this area, it's the gold standard for clinical measurement of brain volume. Not necessarily research measurement, because things like FreeSurfer and other software are used more commonly in research because they're free. But if you want to order it on your patient, you can't order a FreeSurfer scan on your patient. Not really. It'd be very, very hard to do. You'd need to go to a specialized center. But you can order a NeuroQuant brain volume analysis on pretty much any patient, just about anywhere in the country, using typically the local MRI scanner. And this is what NeuroQuant does. This is a coronal slice through the brain. I imagine this as if the patient is facing me, and that would be her left ear and her right ear. And NeuroQuant will take this image and turn it into one that looks like this, coloring the different brain subregions and also measuring their volume. And this is a similar picture, just with a higher resolution, 3 Tesla. That's a magnet strength, relatively high strength magnet, giving us good MRI with good resolution. Beautiful images that we use nowadays. And we always inspect these for the segmentation quality to make sure the regions were identified correctly. And if not, we won't use the volume data. Now, NeuroQuant not only identifies the brain regions, but it compares them to a normal control group. This is one of several NeuroQuant reports available nowadays. This happens to be in one of our patients with chronic mild traumatic brain injury. And I'm going to zoom in on the data down here. And you can see the blue regions are regions which are abnormally large, and the red regions are regions which are abnormally small. And by the way, this is a fairly typical looking NeuroQuant report in one of my patients, where we see a lot of enlargement and a little bit of atrophy. And what these numbers tell you, for each brain region, as you can see over here, you've got left side and right sided volumes. And these are actually the normative percentiles associated with the volume. You have to be above the 95th normative percentile to be abnormally large. So for example, cerebellar white matter on the left and right, and total, which is left and right added together, all are at the 99th normative percentile, very large, larger than 99 out of 100 normal people. So for this, the latest phase of our research that I want to tell you about today, we studied 50 of our patients with chronic, mild or moderate traumatic brain injury, all outpatients from our clinic here. We also had 80 normal control patients participants, they're normals. We got them from the Alzheimer's Disease Neuroimaging Initiative, abbreviated as ADNI study, a really good multi-site national study. There have been dozens of MRI studies using the normals as well as the Alzheimer's patients in that study. And NeuroQuant nowadays measures many brain regions. This is a good approximation of the cortical brain regions from a lateral view of the left side of the brain and the medial view of the left side of the brain. NeuroQuant also measures a lot of subcortical regions not shown here, like basal ganglia, cerebellum, brainstem, etc. So it's a very rich, detailed analysis of brain volume. And Our primary normal control database for this study was the NeuroQuant's normal control database, because that database has thousands of normal controls over the lifespan from ages 3 to 100. And so it's a very valuable source of normal control data. So we had two types of normal controls, the NeuroQuant normal controls and our own ADNI controls, which are the smaller group of 80. We use them for different purposes, but just keep in mind the primary normals were the NeuroQuant normals here. And one big advantage of the NeuroQuant normals is that the data are adjusted for intracranial volume, they're matched for age, and they're matched for sex. Big advantage, because all of these variables can affect brain volume. We studied the NeuroQuant's test-retest reliability for many brain regions and found that they were all excellent, had excellent test-retest reliability. Most of them in the 0.9s and higher, all of them were at least 0.78 or higher. Whoops. And then what we did for the study, one of the main things that we did was to compare the patient's brain volume values to the NeuroQuant normal controls brain volume using the patient's Z-scores. And the Z-scores were taken from the NeuroQuant reports, like the one I showed you with the normative percentiles, which are adjusted for intracranial volume, matched for age and sex. And remember, that report had normative percentiles, which you can see here, and we took those numbers, and for any normative percentile, you can figure out the Z-score, which is how many standard deviations away from the mean is that value. Here, for example, at the 5th normative percentile, that's going to be between -2 and -1 Z-scores or standard deviation units. It actually happens to be 1.64 standard deviation units or Z-score. And if you're greater than, or I guess more negative than -1.64, you're going to have abnormally small volume. You're going to be in the lower 5 percentile, the tail of this normal distribution curve. So that's how we got to the Z-scores. And this is just the equation for the Z-score if you're interested. It's how far away is your patient's volume data from the mean of the normals relative to the standard deviation of the normals. And then we used Z-test to compare the patient's volume to the NeuroQuant normal. Z-test is similar to a t-test. It's a basic statistical test for comparing two groups. But unlike a t-test, for the normals, we entered into the statistical program that we expected a mean of 0 and a standard deviation of 1, because basically the NeuroQuant system was set up that way. And that would equate to a mean of 50th normative percentile and a standard deviation of 1.64. Well, standard deviation, never mind. It's 1.64 would be the Z-score that correlates with a percentile of 5%. So anyway, here are the results of the Z-test. I'm going to go through these a little bit quickly because I'm going to summarize them in the end, but just to show you, if you're interested, we do have a lot of the details. And we've got the means and standard deviations for the patients. For the normals, like I said, it's going to be a mean of 0 and a standard deviation of 1. The Z-statistic and the p-value associated with the Z-statistic. In the column on the right, we have the effect size, D, Cohen's effect size, D. And here's the scale for that, from small, medium to large. And if the values were associated with abnormal enlargement, I colored them in green. And if they're associated with atrophy, I colored them in red. You can see here, there's a lot of enlargement. Cortical gray matter, which is constitutes about 30% of the brain. That's all the cerebral cortical gyral gray matter regions. Cerebellar white matter, very large effect size here. Some subcortical regions are abnormally large. Caudate abnormally large. Pallidum went the opposite direction, abnormally small. The cingulate had several abnormally large regions. Then we get into the frontal lobes, some abnormally large regions, although the orbital frontal regions were abnormally small, maybe because of more injury in that part of the brain where the skull ridges at the bottom are sharp and it may cause more damage than other regions might get. That's just a hypothesis. The parietal lobes are almost all enlarged. Occipital lobes had some enlargement. And finally, the temporal lobes had some enlargement. There's a little bit of atrophy there in the inferior temporal area. And entorhinal enlarged and amygdala and hippocampus abnormally enlarged. And this is to summarize the data I just showed you. And you can see overall a lot of abnormal enlargement, a little bit of atrophy. And when I first started seeing this sort of data, I was surprised because I didn't expect to see abnormal enlargement. I had probably never read about that in traumatic brain injury. And for quite some time, I didn't think it was due to TBI because I just didn't think that would happen. But after it's been a couple years collecting this data and then now the group analyses and for other reasons that I'll tell you about, I think it's probably true, but you can let me know what you think. We were interested in plotting graphs of the change of volume over time. Now this is all cross-sectional data, so it's really a plot of the brain volume versus time after injury. But it can give us an idea, I think, of how the volume may change over time. Here's a cortical gray matter volume. Remember, one of the big brain regions. We're plotting the z-scores versus time after injury in months. And you can see, and at 0, I did not have actual data here, but I assumed that because these patients had were screened to exclude any problems which would cause brain volume abnormalities to be entered into the study, we can assume their volume should have been normal or very close to normal before they had their brain injury. And that's why I assumed they were 0 here, and I connected the line to 0, z-score of 0, which would be the average for normals. And then, you know, the black dots give you the individual patient data, and then these red boxes here are an average of every 10 patients over time. I just averaged them up, and we drew a line connecting the boxes. And then here are the standard errors of the means, the bars here, to give you some idea of the variability of the data. And it appeared to me that there's a very rapid increase of volume to an effect size of over 0.5, which is, you know, it's medium to large effect size there with the z-scores that big. And it more or less plateaued here, actually the whole, you know, out to almost 40 months on average. For cerebral white matter volume, there's a decrease of volume. It went in the opposite direction. And by the way, I forgot to mention this, but I do want to mention this. Am I going the right way? Yeah. Notice for cerebral white matter, there was not a statistically significant difference in volumes between patients and normals. The effect size is minus 0.23, which is small, and the p-value associated with it was 0.12. So it was leaning in that direction, but didn't make it. And for most studies, most things I probably wouldn't have said or thought more about it, but we know in traumatic brain injury, there's a lot of cerebral white matter atrophy and from other studies, mostly in moderate to severe TBI, but even some in mild TBI, some studies. And so I'm going to build a case for cerebral white matter being involved here. And my next step in the argument is this graph, because here you see the relatively rapid atrophy down to moderate effect size of minus 0.5. Then it returns back toward normal. And based on this graph, I did an exploratory analysis and looked at the 20 patients closest in time to injury. And they did have statistically significant abnormally small cerebral white matter with a p of 0.05 and an effect size of minus 0.5, which is moderate. So I think there probably is something abnormal happening in our patients with the cerebral white matter. I think it may be confined to the first 10 months or so of injury. But, you know, take it with a grain of salt. We'll talk more about that because there's some other data related to that. And actually, I'll tell you some of that right now. Cerebral white matter volume correlated significantly with volume of regions which were abnormally enlarged, the ones I just told you about a minute ago on that summary slide. So the primary motor cortex, which is abnormally large, correlated significantly with cerebral white matter volume. in the direction you would expect. So abnormally large motor cortex correlated with abnormally small cerebral white matter volume, giving you a negative value of your Pearson R correlation there. Same kind of thing with temporal lobes are abnormally large, correlated with smaller white volume. Posterior superior temporal sulcus region, same thing. Fusiform gyrus in the temporal lobe, same thing. So there are more correlations than we would have expected by chance alone. This is another way of signifying that there is something abnormal about cerebral white matter volume. Moving on to cerebellar white matter volume, which had one of the biggest effect sizes, you can see there's very rapid enlargement that may go for more than the first 3 months, maybe out to 8 months or so. Hard to tell because we don't have a ton of data here at each of these divisions of time. Then it seems to trend down over this period of time. Doesn't get all the way back to normal, but gets maybe almost a little more than halfway there. Hippocampus also had a big effect size. Here we get very rapid volume enlargement, which goes steadily down over time. And then I was looking for a composite measure of volume because I wanted to look at how it changed over time. So to do that, I needed to use our NeuroGage normal controls. And I took our 80 normal controls. I picked out the 55 that had the lowest years of education so that it matched the education of the TBI patients, because education can affect brain volume. And then, by the way, I did a secondary analysis and just simply compared volumes between these two groups. And the pattern of abnormal volumes was fairly similar, not exactly the same, but fairly similar to the pattern we saw when we used the NeuroGage normal controls to compare volume to the patients. That was the primary analysis. Anyway, I then took these NeuroGage normals and did a standard least squares regression using the patient data, the NeuroGage normal data, and I asked the regression model basically to tell me, give me one composite value based on these volumes that would best distinguish patients from normals. And then I saved that value as predictor z-scores, as predictor scores, and I use z-scores to do my next analysis. And so here's the graph of the predictor z-score that best distinguish patients from normals, again plotting it versus time after injury. And I think of this as a kind of a composite of the previous plots with everything going in a positive direction. So here we see very rapid increase of the predictor score in the first 2 to 3 months, and it roughly plateaus and stays plateaued over time. And now remember, all those enlargements would contribute to this pattern, but the cerebral white matter atrophy also contributes to this pattern because that's entered into the regression equation with a negative coefficient so that all the signs line up the same way. That's why we get these giant effect scores here of like 2 and 3, which are very, very large. And now if we zoom in on this part right here in the next slide, this is that very rapid enlargement, which I've stretched out the time axis here. This is the first 10 months after injury. And I want to point out a couple things. First of all, most of that rapid enlargement seems to occur within about the first 3 months. If you try to look at this individual data here, you see we have very, very little data under 3 or 2, almost no data under 2 months. And so we really don't have much detail with what happens between time 0 and this, let's say this dot here. And we just drew that line by interpolation. It could have been the blue line I outlined here. Maybe it went up even faster, maybe in a month or 2 weeks. Or maybe it went up some other way. We don't know. The only way to know would be to get more data in here, and we would very much like to do that. And if anybody gets patients close to the time of injury and wanted to work with us, we can squeeze those patients in often. We have a mechanism here to do that in our schedule. Could see them quickly, order an MRI, and later NeuroQuant it, and get some volume data down there. So if you're interested in possibly collaborating on that, let me know. You know, this is, we've been doing this 8 years, and this is how many patients we've got that close to injury. Very hard in our setting to get them much more quickly. Even that we've tried and we've gotten them closer to injury, but still hard to break down below about 2 or 1 month. Okay. Now, you know, if you're wondering, well, can the brain really enlarge that fast within 2 or 3 months after injury? I think it's possible. Here's a couple classic studies in neuroscience you may be aware of. One were the syndactyly study by Mogilner et al. In that study, people had syndactyly where their fingers are stuck together or webbed. And they did surgery and opened up the fingers. Before surgery, on the primary motor cortex, the areas for the different fingers tend to be closer together than would be normal, kind of merged together, similar to the way the hand is merged together. After surgery, 26 days later, this is using magnetoencephalography, the finger regions spread apart and look very much like normal finger regions. And that all happened, you know, in 26 days. So that can happen pretty quickly, this rewiring of the brain. The phantom limb studies also found that within a few weeks you can have rapid rewiring of the brain after amputation of limbs. You know, then I wanted to look at the literature because I was worried about seeing all this abnormal enlargement. And, you know, I think most of you are researchers, and if you're like me, you'd want to have something that's the first finding and something that might be important, but at the same time, you're going to worry you did something wrong and you'll be very embarrassed later. So I went to the literature to look, did anyone else find any abnormal enlargement in the same kind of patients we have, chronic, mild to moderate traumatic brain injury patients. There are about 12 studies now, a little bit more than I realized going into this. To make it easier on you, I'm going to highlight the ones that found atrophy of the brain and those that found enlargement or thickening, cortical gyral thickening, which should give you volume enlargement. And I highlight those in green. And you can see in the literature, these studies didn't find any volume abnormalities. These found a mixture. There's more atrophy than enlargement, but there is a little bit of enlargement. It's not zero, like I might have thought a little while back. One of these is our own study right here, which had a subsample of our current patients. So that's not a completely independent study. But it is, our study today uses somewhat different methods, so it's a partial replication of some of our earlier findings. There are a couple other studies, Wang et al. And Govindarajan et al. Found some abnormal enlargement. So what we did next was to look at whether these volume findings of these studies agreed with ours, which means that they'd highlighted in yellow, or if they disagree, that means found the opposite, then they're going to be in aqua. And if they didn't find the same or the opposite, I just left them without any highlighting here. And you can see the yellow ones show agreement, and there are several of those, whether it's atrophy findings or abnormal enlargement findings. We do have some support in the literature for several of our findings. On the other hand, there's also a lot of aqua where they found the opposite of what we found. I cannot easily explain that. There are, you know, many differences in these studies between the exact type of patient you have and you know, how long after illness and the types of brain imaging and volume measurement techniques you use, etc., that could possibly explain it. But I thought this was enough data that I felt, well, you know, we're not really the first to find enlargement. This may be a real finding. So I'm going to go with that and see what else we can get out of this data. Now we're going to talk about asymmetry. Up to this point, we've talked mainly about volume. And asymmetry is how big is one side of the brain versus the other side. Here you can see a coronal MRI of one of our patients who had chronic mild to moderate traumatic brain injury. And you can just look at this image and see that the left lateral ventricle is bigger than the right. And we looked at several slices before and after this, and through most of the brain, the left lateral ventricle was much bigger than the right. We had 3 radiologists look at this MRI or a couple others very close in time, and not one of them said that she had abnormal asymmetry of the lateral ventricles, interestingly. And if you look at the NeuroQuant version, it may be a little easier to see the hippocampus here, which was also asymmetric. The left one was so small in this slice you can't even see it, or you can barely see it. In other slices it was much smaller than on the right. And we measured the volumes and the asymmetries of these regions and found that they were very highly statistically significantly abnormally asymmetric. And we use a measurement of asymmetry, which is the same one that NeuroQuant uses, which is the left side of the brain minus the right side over the mean of the left and right side. And the way I think about this is the mean of the left and right side basically is our best estimate of what the volume of each structure should have been before injury. The theory is that if one of them is injured, let's say the right one, and let's say it shrinks, and then you measure them after injury and you don't know what the volume was beforehand, well, you know, you can estimate that the volume should have been about the average of the two. That's the best you can do. And then you can see the difference between them is fairly big relative to that estimate. That's how, you know, it's a good way to measure asymmetry, and that's what we did. And we use NeuroGage, our own software, to measure asymmetry and compare them to our own NeuroGage normal controls for that. Because NeuroQuant doesn't compare the asymmetry measures to normal controls. And just more generally speaking, NeuroGage, our software, is built on top of NeuroQuant. So we always do NeuroQuant first. We take the volumes that are output from that, put them in our own software, and do some additional analyses and compare them to our own controls. And that's what we did with asymmetry. Here's an example of a NeuroGage asymmetry report. We've got many brain regions. We've got left side and right side expressed as a percentage of intracranial volume, which corrects for skull size. Here's the asymmetry index measured, as I just mentioned. And then finally, the normative percentile, which tells you if these asymmetry indices are abnormally large or abnormally small. That means leaning toward left bigger than right, or leaning toward right bigger than left. And in this example here, you can see several areas of abnormal asymmetry highlighted. And by the way, we did an earlier study where we compared the ability of NeuroQuant and NeuroGage to detect asymmetry and other volume abnormalities versus those of the attending radiologists using the traditional method of routine visual inspection. And we found that NeuroQuant and NeuroGage were much more sensitive, finding volume abnormalities in 96% of our patients versus the radiologists who found them only 13% of the time. Huge difference, highly statistically significantly different. So machine beat man for this study. That doesn't mean we don't want to use radiologists. On the contrary, they're extremely good at what they do with pattern recognition, etc. But I do think that measuring volume would be a great complement to what the radiologists do. Even when you're trained at looking at it and then measuring it, which I've been doing for many years, with medium to large volume abnormalities, you can often see them just looking at the image, but for mild to moderate volume abnormalities, usually you can't just see it, you got to measure it. So we compared our patients to the NeuroGage normal controls with respect to asymmetry. The asymmetry measures were converted to z-scores, and t-tests were used to compare the groups. And we found multiple regions with abnormal asymmetry. Here's cortical gray matter is abnormally asymmetric, and this would be with left side smaller than you expect based on the right. Cerebellar gray matter asymmetric, right side smaller than right side smaller than expect based on the left. That's why it's colored a different color. Before I go to the next slide, we also found one region that had a larger standard deviation, statistically significantly larger, and that survived bone-Ferroni corrections. That's the superior lateral ventricles, which is all the lateral ventricles except the inferior lateral ventricles as defined by NeuroQuant. And that means some patients had abnormally large lateral ventricles and some patients had abnormally small superior lateral ventricles. Looking at other regions, we see a couple more asymmetries kind of scattered through the brain, with one side being bigger than you'd expect based on the size of the other. And we'll come back to asymmetry. But for the, I'm checking my time here, and for the last part of the talk, we're going to talk about why do the brain regions enlarge if I've convinced you that they do, or that there's a good chance that they do, at least in some patients. And there are two hypotheses that I and my group have come up with that seem the most viable. One is inflammation leading to edema. And sometimes in traumatic brain injury, you can get chronic neuroinflammation. The second hypothesis is compensatory hyperactivity leading to hypertrophy, which is enlargement. The idea there is that the brain area is working harder because it's been injured, and that by working harder, it gets bigger. And there are various studies in the literature showing when your brain works harder, it gets bigger, like the London taxi cab driver study and the piano player PET scan study and things like that. Now, as we think about it, if there's ways to test these two hypotheses, let me give you an example which helped us pull together these ideas and these findings. And sometimes a single example can help us understand it, in addition to the group analyses. This is a patient of mine who actually had moderate traumatic brain injury from a motor vehicle accident. Most of our patients have mild, but about 10% have moderate. And with acute imaging, this is his CT scan, the typical axial section, left side of his brain and the right side of his brain. I think of this as him lying on the table with the feet coming out of the screen toward us. And you can see there's a lot of intraparenchymal and subarachnoid hemorrhage here. These white areas here and subarachnoid hemorrhage here. This should look dark, like these areas here in the sulci and there should be dark. Instead, they're whitish, and that's blood. That's acute bleeding. They kept him in the hospital for a couple days and make sure that went down. It did go down. He didn't need surgery, fortunately. This is his MRI scan 1 year and 8 months later. On simple visual inspection, at about the same level of the brain, it actually looked pretty good. Surprisingly, I thought we might see a lot of atrophy here. We couldn't see it just looking at it. I couldn't with the ability in my eye. Here I put them on the same screen for you so you can see them both at the same time. Makes it a little bit smaller and pointed out some different landmarks and findings. But didn't look that bad 1 year and 8 months later. However, when we measured the volume, we saw many, many brain volume abnormalities on the NeuroQuant triage analysis. The first big thing that pops out is multiple areas of abnormal enlargement. That's all the blue color. But there is a couple areas of atrophy, the red color. And in fact, here you go. We do see abnormally small cerebral white matter on the left in this patient. And although this is just one out of the 2 little red areas, this actually is about 25 to 30% of brain volume. That's a really big part of your brain. So that's a big part of his brain is abnormally small, despite all the blues on the pattern here. So then we got a lot of blues, both subcortically and especially cortical gyral regions. Next thing we'll notice is if you look at the left side of the brain, this column here and here, there aren't as many abnormally large regions as the right side of the brain, which is this column here. There are at least twice as many regions on the right which are abnormally large. But we wanted to take that a step further and actually measure the asymmetries and were they abnormally asymmetric. So here's our NeuroGage report, and yes, actually there were quite a few abnormally asymmetric regions. You can see whole brain is abnormally asymmetric, for example, with the left side of the brain being about 4% smaller than the right. Similar with forebrain parenchyma, which is all the brain except for brainstem and cerebellum, as defined by NeuroQuant. Cerebral white matter, highly asymmetric, 9% smaller on the left and right. That's less than the 0.001 percentile. That's like 1 out of 1,000 or 5 or less, even very, very small. And that was a general pattern. The left-sided brain regions tended to be smaller than the right. Not all of them, but the majority of them. Here's a picture of a couple that you can see pretty well on this axial section. You can see the left hippocampus smaller than the right, and the left inferior lateral ventricle bigger than the right. And if you look at the slices above and below it, it looked similarly. And when you look at the numbers, left hippocampus abnormally symmetric, left smaller than right, and inferior lateral ventricle abnormally asymmetric, left bigger than right. So the numbers match the pictures here. And it also reminds us that if a brain region shrinks, the ventricle next to it will get bigger to take up the extra space in the brain. The opposite is true also. If a brain region enlarges, the ventricle next to it will shrink smaller to allow everything to fit in the brain. And so just looking at this doesn't really tell you what got bigger and what got smaller, but we'll discuss that in the next few slides. So to summarize this case, on the day of the accident, he had left-sided cerebral hemorrhage. That tells me he had more traumatic forces to the left side of the brain. That's why he had bleeding on the left side, but not the right. A year and 8 months later, he's got a lot of less cerebral white matter atrophy on the left. He didn't have atrophy on the right. Again, it seemed to be more forceful injury on the left. He also had widespread enlargement both sides of the brain, but it was more on the right, interestingly, the opposite side from where we thought he had really more forceful injury. Consistent with that, he had a lot of asymmetry with right bigger than left, or to say it equally, left smaller than right. So why were his brain regions enlarged? Well, I think that, first of all, if the inflammation hypothesis were true, that would predict that left-sided injury would lead to more inflammation and swelling on the left, that the left side would get enlarged. We did not see that. I don't think his case supported that hypothesis. On the other hand, the compensatory hypertrophy hypothesis would explain why the left-sided injury led to right-sided enlargement. And that would be because you had compensation of contralateral brain regions. To put it graphically here, before injury, we've got normal cerebral white matter volume, normal volume of the other brain regions. After injury, the white matter is injured and atrophied, and other brain regions that are less injured, maybe on the other side of the brain, get bigger as they work harder to try to compensate. That's what I think his case was supporting. And then in the last final stage of the talk here, I want to talk about some group hypotheses we did to look at, analysis we did with group data to look at the two same hypotheses. So the inflammation hypothesis would say, in terms of what would you see with asymmetry, that's where we're going here. It would say that, let's say if one side of the brain is not injured or less injured, you'd have pretty normal volume. The other side of the brain is injured. The white matter would atrophy, the long, thin white matter tracts that we know from a lot of literature, often as damage, diffuse axonal injury and traumatic brain injury. And then regions close to that would hypertrophy. That's what I think the inflammation hypothesis would state, because they're closer to the damaged regions. And so if you looked at the asymmetry correlations within the group of patients, I think you would get negative asymmetry correlations, and we'll come back to that in a minute. On the other hand, with the compensatory hypertrophy hypothesis, we would see the opposite. It would explain the opposite. So you've got injury on one side of the brain here with white matter atrophy, but less injury of gray matter regions. But on the other side of the brain, the contralateral regions here that are basically normal or much less injured are compensating for the contralateral regions, which may be partly injured. In contrast, the cerebral white matter volume here would probably stay normal. So and then when you look at correlations within the group of patients between cerebral white matter asymmetry and the asymmetry of enlarged regions, you would predict to see positive asymmetry correlations. And let me explain it a little bit better because I didn't explain that well enough. So we're going to look at asymmetry correlations between cerebral white matter asymmetry and the asymmetry of the regions that were abnormally enlarged, which we found from the earlier phase of our study, just doing simple, straightforward volume analysis. Remember, there are multiple abnormally enlarged regions. And we're going to look at asymmetry correlations. And here you can see when the cerebral white matter is atrophied, you're going to get left side smaller than right. You can see this white matter area is smaller than this white matter area. With the enlarged regions, you're going to get right side bigger than left. You can see this right-sided region is bigger than the left. And if you think about it in terms of asymmetry, right greater than left is exactly the same as left smaller than right. They're equivalent in terms of the direction of the asymmetries. They should correlate together. And you have the opposite situation here. But we could look at that, so we did look at that, and we found actually several positive correlations. First of all, for cortical gray matter, the asymmetry correlations with cerebral white matter was in the same direction and significant. Same with several other regions that were found to be abnormally large. They correlated significantly with, their asymmetries correlated significantly with cerebral white matter asymmetry in the expected direction. So our group data also supported the compensatory hypertrophy hypothesis. And then in conclusion, we found enlargement of many brain regions in our patients with chronic mild or moderate traumatic brain injury. We found atrophy of a few regions, including cerebral white matter, although that may have been limited to the first 10 months or so of injury. We found a lot of abnormal asymmetry in our patients. And then trying to kind of tie this together in one final graph here, before injury, both sides of the brain have similar volume, normal asymmetries, fairly symmetric. After injury, we have atrophy of white matter, we have enlargement of other brain regions that are less injured, and we have the development of asymmetries as a result that occur on these sides of the brain. So that's the end of my talk. This is our team here, and I want to thank them a lot for all the help. They've been a great help. So, many of you may know Dr. Al Oakes, my colleague and friend, and he's been my mentor since I was a medical student back at VCU many years ago, back when Dr. Merchant also was one of my professors. So it's nice to come full circle all these years later with this work. These are some of our other collaborators, particularly Dr. Bigler, a prominent researcher in this area, and we've been very fortunate to have him as a collaborator and mentor of ours as well. And many students, we have many students rotate here. Tell your students if they're interested. We teach them from college level, med school residency, and older. They see patients with us, do research with us, learn a little bit about running a small business because we are a small business, and we really enjoy having our students. This is my contact information. I would be happy to get any feedback, questions, or comments that you have by email or phone. Right after this meeting, if you happen to be watching live, we're going to be discussing it with Dr. Oakes and John Seabaugh, and if you feel like calling us on the phone, give us a call and we'll be glad to talk with you about it. And I'm just going to switch back to my last, yeah, there. I think you can see me now, and I will sign off at this point. Thanks for listening.